between functions.
Okay, thanks. Thanks, Eric, for the invitation. Thanks, everyone, for listening. And this talk is going to be a little bit different, I feel, anyway, from the talks this morning. Definitely. Yes, it will be 40 minutes long, or possibly even less than 40 minutes long. And it's also much more on the application side. So feel free to ask questions. I have given this talk in front of different audiences, but it's really hard for me to judge how much people know or don't know, and also what bits are obvious or easy to understand, hard to understand, whatever. So please interrupt and ask questions. Oh, wow.
I didn't tell him to tell you. You can do the slides by hand and just as the...
Yeah. I can what? I can... It's very closely correlated with you shutting that door. I was like...
If this is this, maybe you...
Yeah, I could take this out, but I don't think it was this, because this was in already when it happened.
Ah.
I mean... Oh, that was good. Okay, great. And then I just put... So you just unplugged it and put it back in?
Yeah.
Okay, cool. So next time I can... Okay. We'll try. Alright, so I'm going to talk about... basically I'm going to talk about an inverse problem, but it's an inverse problem that involves protein sequence data. So this isn't quantitative data. I don't have measurements. All I have is protein sequences. But I'd like to use those sequences, and we have more and more sequences. We have experimental technologies that make it extremely easy to collect sequences now. So we have millions of these sequences. I'd like to be able to use the sequences to make inferences about protein structure and function. Okay? So a single protein proteins are made up of amino acids. These are like the building blocks. There are 20 different amino acids in nature. Right? So a protein is literally a string. It starts life as a linear string of amino acids, and they're joined together by peptide bonds. There might be about 300 amino acids in a protein, roughly. But basically, these amino acids, they sort of dictate the properties of the protein. Right? There's some kind of coding or mapping from the space of these 20 molecules, which get joined together, to this sort of infinite variety of proteins that we see in nature. Okay? So we now have samples of the sequence of amino acids that corresponds to a particular protein you might be interested in from lots of different species. So it's like there's hemoglobin in me, there's hemoglobin in chimps, there's hemoglobin in crocodiles, there's hemoglobin in all sorts of animals. Those hemoglobin molecules are sort of similar to each other. They fold up in roughly the same way, they have roughly the same structure, they have more or less the same function. But there are differences in those sequences, right? So my hemoglobin sequence might be only 2 positions or 2 amino acids different to a chimp's hemoglobin sequence, but it might be almost entirely different to a crocodile hemoglobin sequence. So those different sequences somehow represent samples from the probability distribution on the space of sequences, and those samples all correspond to this protein, hemoglobin, right? So that suggests that we should be able to make a probability model on the space of amino acid sequences that somehow describes which sequences correspond to hemoglobin. Yeah, there was a noise, definitely. Okay, so that's the idea. That's what I'd like to do. So basically, just to go over, this is kind of a toy picture of how things look. The blue line is the sequence of amino acids. I've highlighted 2 in red spots. Those are 2 different amino acids. We indicate them by letters because we need 20. So these 2 amino acids, D and K, they're charged. They're actually oppositely charged. So this forms a salt bridge. It's a nice interaction. Sort of stabilizes the protein. If you denature this protein by putting it in some kind of chemical denaturant, it unfolds and it stops working, so we call it inactive. But then if you take it out of that denaturant and put it back in physiological conditions, it refolds and it regains its function. So really all the information is contained in that sequence of amino acids, at least for most proteins. So between different species or different individuals, you get mutations in sequences, right? So in this case, for example, that D might change to an H, it's a different type of amino acid, and it has a positive charge. The D used to have a negative charge. So now this charge interaction, this charge pairing is sort of broken, right? And that might cause the protein to no longer fold up properly, to become inactive. So let's go back. But you might imagine that that could actually be rescued. So this sort of disabled protein that doesn't work might be rescued if another mutation occurred at that second site. So that K changes to a negatively charged amino acid, so you have that salt bridge back again. Okay? So the idea is that somehow there might be correlations between the mutations that are accepted at different positions. In other words, different positions might not evolve independently in a protein. So I'd like to understand... Yeah?
On your little cartoon,
yeah.
that little scribble is
Yeah.
exactly the same as that one, but that
Yeah.
it's like it has to be something similar but not identical. Or
No, it could be identical, right? So I mean, these could have exactly the same structure, they both work, but you either see KD at these two positions in the protein, or you see DH. And so the variables are going to be those positions in the proteins, right? So you never see the intermediate. So you have this perfect correlation between these two positions. It's either KD or DH. You never see KH in your sequence alignment, the intermediate. Okay? And so yeah, I want to understand how the different mutations at different sites.
So
Yeah.
you mean that it's not invertible, in the sense that the amino acid sequence specifies the trig structure, but you have two trig structures with very different sequences.
Yeah. And in fact, I'm actually going to have lots and lots and lots of sequences which all have the same structure. Right? That's exactly what I'm hoping to. That's the data I have, essentially, yeah.
How do these mutations occur? Is it spontaneous, or is it
Yeah.
triggered by something?
So it could be spontaneous, it could be triggered by something. Basically, in evolution, what we believe happens is that mutations are randomly generated, and they're either accepted in the sense that they become prevalent in a population. So if a mutation makes an individual more successful somehow at producing offspring in particular, then that sequence will spread. And so
how do you define the family?
So we define a family by proteins which have similar structures. When I say similar, I mean the same. Essentially the same structure and function. And so we define that using existing models of similarity between sequences. So we have these large alignments, basically. So we have a large set of sequences which are different to each other but correspond to a protein with the same structure and function. So it's really a sampling of the space of possible sequences that makes that protein.
So you are suggesting that to determine the chemistry of a protein is not of much interest.
Is not what, sorry?
Not of much interest, in the sense
Oh,
many different
No.
chemistries.
What I'm asking is, can we use the variation or the correlations in those sequences to make predictions about the protein?
How do you get
But you define
this is a K and a D, that's a D and an H.
Right.
maybe there's a pair that's neither a K nor a D that would go in. So what's your criterion for deciding that it's the same protein?
Well, essentially, it looks the same and it does the same job.
So there are tests of functionality.
There are tests of functionality, and we also can predict functionality pretty well just based on sequence similarity. But it's an important point. We can't predict it perfectly.
But you can have sequences that are actually quite different.
Yes.
Still say that's the same protein
Yes.
based on functional characteristics.
Based on functional characterization, and also we have models which sort of some parts of the sequence being different are more important than others, right? I mean, there's a whole field of this basically, but we have these large sets of sequences. That's like our starting data. Yeah. So we have lots of genomic sequences, lots and lots, right? We'd like to know about the space of sequences that corresponds. Oh, I keep pressing the wrong button. Sorry. That corresponds to a particular protein, right? So in this example, we might end up with this sequence alignment. So these are toy sequences from different species. This is human, mouse, I think this is cow and horse or something, right? So in these sequences, you either see a D and a K, right, or an H and a D. So the variables in general are going to be these columns, the positions of an amino acid in a sequence, right? And so here, I would say that there's a correlation between column 1 and column 6 in my sequence alignment. Okay? No idea. Right.
You have this miracle of the resurrection. So
Pardon?
you have the resurrection, so you
Yes,
can stay
yeah,
down.
so that's fine. So basically,
Can we go back to what you mean the
Yeah.
correlation between it's human and mouse, is it?
no, I mean between the first column
Yeah.
and the 6th column. Right? So whenever the first column changes,
Each row is
Okay.
a protein, right?
yeah, each row is a protein, and I'm looking at the correlation structure between the columns. Right?
The first column changes, the sixth column changes in,
Yeah.
in a predictable way.
Yeah, or at the same time.
Yeah.
I'm not so worried about the identity of the amino acids involved, just that the changes happen at the same time.
And every line is just a sequence that gives a protein of same shape and functionality.
Yeah.
Right.
Yeah, exactly, yeah. Right, so I'm going to have this hypothesis that interactions between amino acids constrain sequence. They constrain how that sequence can change, right? So this is a contact in the 3D structure, there's 2 red amino acids, and those 2 red amino acids change at the same points in the sequence alignment, right? Of course, what I'd like to do is I'd like to start from the sequence alignment, look at the correlations, right, and make predictions about amino acids in contact in the structure. So essentially I want to use statistics of a multiple sequence alignment to make inferences or predictions about protein structure and function. Okay? So the first thing I need to do is I need to somehow measure the correlation between every pair of columns in the sequence alignment. Right? So here I have all of the columns against all of the columns. I have a little matrix of correlation scores. I need to somehow measure these correlations. I'm going to consider the column pair frequencies. So how often does the pair K and D occur compared to how often does K and how often does D occur, right? It's a very simple statistical kind of measure. One thing I might use is mutual information. So I can compute the mutual information for each pair of columns. So if I do that, do I end up with high scoring residue pairs? By residue pairs, I mean like, 1 and 6 here, corresponding to contacts in 3D. Well, that's a very simple question. We can test that very easily. This is a map of a protein structure. So this is experimental data, right? So this is, when I say map, it's basically a binary matrix, right? These are all the amino acids in the protein. In this case, it's like 166 against all the amino acids in the protein. And there's a 1 or a blue spot in this matrix whenever the 2 amino acids make a contact in structure, okay? So you can see that there are 1s all the way along the diagonal because 2 amino acids that are close to each other in the sequence are going to be next to each other in the structure because they're joined, right? And then there are also some 1s off the diagonal, and those are where different sort of parts of that string loop round and touch each other, okay? So that uniquely defines the protein structure, this map. So I'm going to run my mutual information analysis on a large sequence alignment, and I'm going to plot the high-scoring pairs on top of this map, okay? I'm going to put them on in red, and what I'd really like is for the red points to fall on top of the blue points, right? So, as you can see from this picture, that doesn't really happen. It's a sort of a mess, right? There are lots of red points where there weren't blue points underneath, and there are lots of blue points that don't have any red points. So this is no good, okay? But if we look at these red points, you sort of see that there's kind of a block structure to them, right? There are these sort of filled in patches and lines, and you sort of start to wonder, well, why is that? And if you think about it for a few minutes, you sort of realize that once I have a few amino acids in a protein, right? Now here, I mean, K and D are next to each other, so I predict they're going to be correlated, right? That's my hypothesis, right? Well, E and K are also next to each other, so they're going to be correlated. And D and R are next to each other, so they're going to be correlated. But this correlation business, every time the E changes, right, the K changes, these columns in my alignment. Every time the K changes, the D changes, because they're correlated, and every time the D changes, the R changes, right? So the problem is that this correlation is a transitive property, right? So I don't actually just see these correlations, which I sort of want to see. I also see these extra correlations because of this transitivity problem. And that sort of, in my model, ends up with these sort of filled in blocks, which is what I'm sort of seeing in the data. So what I need to do is I need to,
Why do you expect the correlation between D and R and E and K? Just because they're close along the line?
No, because they're close in the structure. So that's my sort of premise,
Oh, it's
hypothesis.
not because they're in the sequence, just another content.
Yes, they're not close. They're not meant to be close in the sequence. They're just
I just misunderstood you.
yeah. No, so they're just close in the structure. So like this.
Yeah.
Yeah, I could have drawn it better. It's a good point.
But wait,
Yeah.
So if that was the case, why wouldn't you expect to have that the red cover all of the blue and more? And you showed that the red is not covering.
I only put on the top 100 red scoring pairs. So I could put more pairs. I mean, I have
And then
scores for every single pair of amino acids. Yeah.
I'm just very
Yep. Yeah, sure. Yeah. Okay.
So just right now, so you are really, the red part, really the covariance matrix.
Mhm.
So you're not doing like precision matrix. What
Yes.
you're usually doing is
More or less. It's not quite a precision matrix because it's not a continuous model, but what I actually do will end up being the same as what you would do for a precision matrix, basically, yeah. Things. My computer doesn't normally do this, I promise. Like, this is not. So, what we need, of course, is a global probability model, right? Because somehow we want to infer the minimal set of interactions that can reproduce all the observed correlations, right? I just want those main sort of red arrows. I don't want these other additional arrows. We're going to constrain our probability model using just, oops, press the wrong button, just the first order and second order statistics of the data. I'm not going to use higher order statistics because I don't have enough data. I don't really even have enough data to use the second order statistics, but I'm going to sort of just go for it. And I'm going to use a maximum entry modeling approach. And that basically means, because this is not continuous data, these variables take one of 20 states, right? So I end up with a Potts model where I have single site terms and I have pairwise interactions. Okay? So this literally gives me a probability model for a sequence, or that a sequence is part of a protein family. And I'm going to fit those interactions using an assumption that the couplings are small. So if there's a parameter in front of this interaction term, it would be small. In fact, you could put an alpha there and just say that alpha is small. So in the limit that the couplings are small, you can approximate the EIJ parameters just by inverting the observed covariance matrix, which is basically, as far as I can tell, very similar to computing a precision matrix. And I'm sort of bemused by that analogy, and I don't know, I'm intrigued by it. I mean, if this was a multivariate Gaussian distribution, of course we compute the inverse covariance matrix. So quite why we need this assumption that the couplings are small. But in statistical physics, that's how people get to this equation. So, of course, our data is undersampled. We're fitting something like 2 million parameters here. We have something like 10,000 sequences. So our covariance matrix tends to be singular. And we basically use shrinkage to perturb the matrix away from being singular so that we can invert it. So we add lambda times the identity to our observed covariance matrix.
But you want this thing to be shrinkage really is you want to make it sparse. Somehow.
Well, shrinkage, I mean, basically we change what's on the diagonal, right? So yes, people use shrinkage and they want something to be sparse. I don't think this has the effect of sparsifying our data.
Because I thought previously you had a covariance matrix that
Yeah.
in fact is giving too much correlation because you
Oh, yeah, but I'm adding lambda I and then I'm inverting, right?
This is precision matrix is a conditional covariance. It's like conditioning of errors and you see the covariance. So it's much more sparse.
But I think that, it will be much more sparse whether you do the shrinkage or not. It's just that I can't invert it.
do you really need to invert it? When you're
Yes.
where you want to go to compound.
Yeah, I really need to invert it. In fact, what I have tried doing is, of course you could estimate the inverse just by taking the outer product expansion, right, and taking just the non-zero eigenvalues. And if I do that, then it doesn't work nearly as well. So this shrinkage is doing something. It's not clear to me that it's sparsifying the data, but it's certainly regularized. It's certainly making the performance better. So it's important. It's not just making it invertible.
Shifting the eigenvalues.
Yes, it's shifting the eigenvalues.
I don't think that you would call that shrinkage. I think you call it ridge regression.
Yeah, you could. In statistics, you would call it ridge regression, exactly. Yeah.
It's not L1, it's L2. Anyway.
Maybe. I don't know. So in different fields, I've discovered that the same thing has different names. So, yeah. But yeah, ridge regression is definitely. Okay. So once we do this, we take the highest scoring pairs and plot them on top of our contact map. We now find that we're doing a much better job of finding the grey spots. Right? So in each case, I've put 100 spots on the plot. I've taken out the points that are close in sequence, just because we know there's a close in structure, right? Because they're connected by peptide bonds. And so the question is, how much information is there in this set of data? Right? I put 100 high-scoring pairs on this contact map. Does this actually contain enough information to describe the protein structure? Right? And we sort of thought about this, and we figured the only way we could tell is empirically. Right? If we literally enforce these red points as distance constraints on a polypeptide, do we end up with the correct protein structure? Okay? So we start with this unfolded polypeptide, and we constrain the distances between the two residues in each pair. And now there's standard sort of biomolecular computing software that does this, because this is the kind of data people get out of NMR experiments. Right? They get information about which residues are close to each other in structure. So we could take exactly that software and just put this information into it and get protein structures out, right? Which is very convenient because, it was difficult enough already, never mind having to write that software. So it turns out that if we do that, we actually get remarkably close to the predicted, or the experimentally solved crystal structures. So this is a small protein, 67 amino acids. This is the crystal structure here, which is available with this code in the database, and this is our predicted structure. Now you can see that there are some bits of it that aren't quite right, like this blue piece here doesn't point in quite the right direction. There's something funny going on with this red bit, but essentially it's less than 3 angstroms RMSD away from the crystal structure, and that's really very high resolution. It's comparable to a crystal structure, right? But we got that just using information from sequences. So essentially we're able to predict that starting from just sequence information. And so this is the protein that I showed you the contact map for. There are 166 amino acids, and this is just over 3 angstroms RMSD from the crystal structure, over 161 residues. This RMSD, of course, scales with the size of the protein, so you expect it to get larger. And again, as you can see, there are some problems, like this helix is bent, and it should be straight. This is really providing a sort of different way of giving us the crystal structure of a protein, or the structure of a protein. And what's interesting is that, of course, when you crystallize a protein, you kind of freeze it, and you expose it to extremely unusual conditions, you dry it out, you do all sorts of horrible things to it, and often it means that it adopts a slightly sort of strange conformation, or a conformation that isn't necessarily the active conformation. So it isn't necessarily clear that these sort of errors in our predictions are actually mean that we're actually different from the true structure of the protein, right? Because these proteins, when they're actually doing their job, they're sort of fluctuating slightly, they they change positions slightly, there's thermal fluctuations, all sorts of things go on, and so this is well within the realm of sort of possibility. I'm very conscious of the time, so please, yeah?
Maybe for later.
No, go on. I mean, now at this point I can stop at any point, I mean.
Okay. Okay. So you say you have a computer program that goes from your nearest neighbor, whatever. Well, you know what I mean, contact points, and then you come up with a structure. I'm sure there's more than one solutions to that. So, but you come up with only one solution.
So
Why don't you have a full PDF there?
we do actually generate an ensemble of structures.
Okay.
You can generate essentially as many as you like. If we generate 10 and then we cluster them, then typically we find that the biggest cluster, or the sort of best cluster, is, or when we choose it automatically, right? We don't look at them all and choose. We have to choose one blindly for it to be prediction, right? But we can. These are chosen blind, right? But essentially, because we're using such under-constrained data, we have topological mirrors. So very often the clusters are kind of mirrors of each other, right? But then it's quite easy to choose because you have kind of handedness of helices and things like that that can help you to tell. I think what we found is that the tightness of the clusters corresponds to how good the predictions are. So it's nice because in a way it provides a way of sort of discriminating.
What you were showing was just the best cluster.
Yeah, these are our predictions. So they're not necessarily the best, although I think they're pretty close to the best because we've got quite, our criteria for choosing are quite good, essentially. But we have to choose blind. In particular, so these are some more, in these, the crystal structures in grey and our predictions are in purple. But then, what we went on to do is to make de novo predictions, right? Because everything I've showed you so far, there's a crystal structure to compare to, right? But of course, what you really want to do is make predictions where there's no crystal structure, okay? And so we started looking at transmembrane proteins because those are extremely hard to crystallize. Experimentally, they normally exist inside a membrane, so they're very hydrophobic, and so to crystallize them, you need to have them in solution. It's very difficult. And also very important, lots of human proteins are transmembrane, and particularly lots of drug targets are transmembrane, so we're very interested in knowing more about them. This is, we modified the algorithm very slightly to work for transmembrane proteins. So you can make a topology prediction for transmembrane proteins of how they are sort of arranged in the membrane, and we can use that to prune our set of predicted pairs, if you like. So we can automatically remove pairs that we predict aren't actually close in structure. So there's some details about that I won't go into in too much depth. It means that this is our input data, these are our pairs, these are how far apart they are in the crystal structure. So we went from this plot to this plot. Oops. Right, so this is very automated, so we're removing pairs that are close in structure as well as pairs that are far apart in structure. Anyway, it means that we're able to predict the structure of transmembrane proteins with extremely high accuracy. So this is 3 angstroms over 200 residues, which then meant we felt confident in looking at proteins for which no crystal structures existed. So this is, I'm going to talk about this protein called adiponectin 1, or adiponectin receptor 1. Right, it's a transmembrane protein for which no homologous structure, so no structure of a protein that's similar in sequence has been predicted, been solved experimentally rather, right? And these proteins are important for, or they're important for lots of human health issues, so they're sort of quite interesting to biologists. So that means that now when we have our high-scoring pairs, there are no grey points on this contact map, right? Because there's no crystal structure data. So there's no sense in which we can tell how well we're doing at predicting these pairs, right? But we're just going to apply exactly the same algorithm, the same software, and enforce these pairs as distance constraints. And this is the predicted structure that we get. So now this is really, we're ranking this blindly. We've got all these 200 or 400 predicted structures that came out of our computing software, and we had to just pick the one that we thought was best. What we can do once we've made that prediction is we can sort of look at how similar it is to all the structures in the database that have been solved, right? And we can ask, is this sort of highly similar to any structure that's already been solved for a different protein. So that might sort of mean we believe in it more. It turns out that, yes, it's just under 5 angstroms from this bacterial protein, which is bacteria rhodopsin. So this is a human protein which is important for binding of adipocytes. This is a bacterial protein. Bacteria don't have any adipocytes. It's a completely different protein, essentially. And what turned out to be the case is that actually this was flipped in the membrane, or would be flipped in the membrane compared to that bacterial protein. So it really is sort of different, even though the structures seem very similar.
What does flipped mean?
So the start of the linear chain, the N terminus, is going to be inside rather than outside the cell. So literally the topology is just, instead of being this way up, it's this way up. So they have a sort of orientation in the membrane. And you can tell the orientation from a structure by looking at the charge gradient along the protein.
But is there a belief that there's some connection between the bacterium?
No.
No.
It seems like it might just be a good structure that biology has kind of found in many different cases, basically, sort of convergently, so we made these predictions for 11 different proteins, and we're sort of waiting with bated breath for people to crystallize them, and we published them, and I guess the other thing that we found is that some of these high-scoring pairs can sort of correspond to pairs of amino acids or pairs of residues that are close between proteins rather than within a single protein. Right? That's what we found in this example here, and this example here. And so I guess what I really need to do now is to understand. I mean, we have this model, right? We've made this model for this probability sequence for this protein. I don't know if that model really means anything. All I've shown you is that the highest scoring, or the highest couplings in that model provide information about structure, right? But sort of a binary version of them provides information about structure. But that's quite compelling. It suggests that model really tells me something about the protein. And I'd like to know, can I really use that model to infer more information? If I look at the actual values of the couplings rather than just if they're high or low, right? Can that tell me more about the protein? I mean, if I simulate that Potts model, can that tell me something about the sort of energy landscape of the protein, right? How far can I push this analogy? So if anyone has any thoughts on that, I'd be really interested. Yeah.
So you're waiting for the experimentalists to crystallize, which, I mean, for transmembrane proteins, it might never happen.
Yeah.
But of course, what you really want to do is, can these structures that you're predicting, are they correlated with the function? Can they use them for biomedical science? But of course, that can be investigated another way, which is you prune the correlations to understand, to predict which are the real contacts. So now you go and mutate those contacts and see if the resulting proteins have a function or not.
Yeah.
And if they don't,
so actually I've done
then
that.
that's your target.
I had a recent experimental collaboration which was published in October, I think, where I did that, or at least a graduate student did that. They mutated the highest scoring pair, and they showed that it destroyed the function of the protein, essentially. So it looks as though these pairs are really the pairs that are important for the protein. They're the pairs that evolution kind of conserves. And I guess another question is, are there higher order interactions which we're sort of approximating with pairs, or are pairs sort of, are pairs the sort of the key point? Is it worth us trying to find groups of pairs that talk to each other? But I think this idea of making mutations absolutely is a way forward. Yeah. But in terms of the modeling, I guess I'd really like to understand if there's some way of measuring how well we're doing. And all I have is these sequences and these structures, and then a whole load of biological data, basically. So it's a little bit complicated. Yeah, thanks, sorry.
So we have bravo for beautiful talk and being on time.
Thanks.
Questions? We have time for questions. Really.
If you included 3 interactions amongst 3, would you get significantly different predictions? I mean, that's a natural thing to try to see whether your truncated model is sufficient to explain what you want to explain, or whether
Yeah, I guess I don't know how to do the parameter inference for including the third three-order terms. So like when I'm inferring the parameters of my model, I just, I remember I do this matrix inversion to get the pairs. I just don't know what I would do for the triplets. I think it's a very sensible
you
question.
could apply the same. You're doing, I'm not sure exactly how you're doing the parameter estimation, but it's maximum likelihood, I guess.
Yeah, maximum entry. I mean, literally, I just take my matrix with observed covariances and I invert it to get pairwise couplings.
Yeah, of course. Because you've got the max entropy principle that tells you to do that.
Yeah.
But you could do something like maximum likelihood with a higher order model.
Yeah. No, I think it would be really a good thing to do. Yeah, I just have too many things to do. That's the problem. I mean, I think it would be great. Like, yeah. Yeah.
There's something that I don't understand why
Uh-huh.
chemistry does not play any role in this game.
Yeah, no, nor do I. I guess what I'm asking is, does this provide information about chemistry? But no, this is just statistics.
Sounds a bit strange.
I could imagine improvements to the algorithm that would use chemistry. So, for example, some of these amino acids are quite similar to each other, so you could reduce the alphabet and then recompute the statistics and so forth. But no, kind of the beauty
In
of this.
certain case I can understand.
Yes.
But
But there's no chemistry.
from your
No,
analysis,
absolutely.
they're all equivalent, and which is very strange.
Yeah, yeah.
Also because from the steric point of view, they are very different. And
Yeah.
that's important.
No, I completely agree. I don't really understand why it works in that sense.
You have no way to check the level of confidence you can attribute to this kind of prediction.
Well, I mean, structurally.
No, if you have to way that people crystallize,
Oh no, but I have those ones for which the structures are known, right? That I showed you to start with,
No.
and those are really very
in
good.
we have an expression: a small bed does not produce the spring. No,
Uh-huh.
I mean, if you do few cases, of course.
Oh no, I mean, it works pretty well. It doesn't work perfectly. Like there are some cases where I don't have enough sequence data, for example, to make a big enough alignment to produce anything that makes any sense. How many sequences?
Yeah, alliance.
5,000, 2,000 maybe.
There's 2,000 different species.
No, 2,000 different sequences. They can be paralogs, like, sort of, but
2,000 for the same
protein.
structure.
Yeah,
300 amino acids,
yeah.
and you're looking at pairwise, so it's not much data.
No, it's not enough data. It's very under sampled, yeah.
Actually, I had a question.
Yeah.
I realized that maybe you should have not understood anything. But I'll ask it anyway. Which is, so you're doing this model by comparing sequences.
Mm-hmm.
So you get an interaction matrix. I didn't get how you can transport that to any given sequence that is not part of the group.
No, I mean,
How do I do that, even in practice?
well, I have a probability model now,
Yes.
right? So I can feed in any sequence I like, and it will give me a score for that sequence.
But
But
I
I
thought that this probability model was for a specific protein or class of protein that belong into a folded structure.
Yes.
And the part I don't understand is that now you're saying I can actually do this
Yeah.
to predict the structure,
Oh well,
something I don't know at all.
No, but I'm predicting a structure for that alignment, right? For that class.
You're still in the
So
same
how can
class.
you go into another class? I thought that at some point.
No, I build a model for each class, right? But it's de novo because I'm starting with sequences and I'm predicting a structure for those sequences.
Okay. But you will not discover a new structure then. They will always be into shapes that you already know.
No, because there are sequences for which we don't know the structure. So I have a lot of sequences, right? I believe because of, I mean, if I use hidden Markov models to make an alignment, right, then above a certain threshold, I believe that all those sequences have pretty much the same structure, right? That's sort of well accepted.
Right, it's because they have the same function, right? I mean, these are
No.
related.
Yeah, but I mean, this is all prediction from sequence, right? It's not cast iron, but I'm pretty sure that the set of sequences has the same structure. I just don't know what that structure is, right? And so this tells you what that structure is.
Okay, we see the latter. I'm not sure.
Okay, yeah, no, that's fine. It's
Sure, I
not.
don't understand.
Yeah, yeah.
Yeah, let me ask a more statistical question.
Yeah.
So I mean, so you have like 300 amino acids here, and your sequence is like 2,000. for 3,000 sequences.
Yes,
So this is, in the statistical language, this is a very high-dimensional problem.
yes.
So do you have any trouble like using the sample covariance matrix to invert it, or
Well, I mean, this
do
is-
some like sparse precision matrix model?
Well, so I mean, we just do this regularization, right? Where essentially we add lambda I to the sample covariance matrix, and lambda is approximately 0.5. I mean, if you vary that lambda a bit, it doesn't actually make very much difference. But yes, we do have problems. We do this very stupid thing, stupid but sort of low, simple thing, and it seems to work. I know that if we do something slightly different, then it changes the results. And it's clearly an important question. Some people published a paper where they used lasso to regularize. That actually didn't seem to make things much better. I was surprised.
You have to check like G-lasso, since this is really a Gaussian graphical model.
Yeah, so I think they used something like G-lasso. This was some people from from UCL, and I was really surprised that it didn't. I thought it would be great, but yeah, I wasn't.
I don't want to be binary, but
Yeah.
structure and function
Uh,
is something mediated by the chemistry again.
yes.
Okay, and you seem to identify them without any problem. So you seem to conclude that the amino acids are just rubbish, in the sense you use them to fill something, and once you have put there, it works. It's very strange.
I completely agree that it's very strange that we
Not
do.
only the statistics, but
Yeah.
also this fact that you identify structure and function. In between the two,
Yeah.
there are the individuality of the chemical points. It is true that more or less the matrix of a protein is just a neutralizing factor, so that the active site, for example, is not ruining the organism. So in this sense, it's not a great function, but at the same time, certain idiosyncrasies of the chemistry there are important. So function and structure should be different.
I mean, this stuff is clearly important, right? But somehow we're able, just starting from the sequence data, to get a sort of a good approximation of at least the structure, right? And also, perhaps we can identify the amino acids that are important for function.
There is
I
no
don't
reason
know.
not to expect that a small difference in structure couldn't produce a large difference in function.
Yeah, that's true, but maybe, I don't think so. I think the differences we're seeing are so small. It's within the resolution of a crystal structure.
I understand that the result is intriguing.
Well, I agree, it's very surprising. That you can get this far without knowing any chemistry, essentially. So.
No, it's a surprise that is motivating me not to.
Yeah. No, I agree. I think you're right to be surprised. Thanks.
What's your threshold for clustering sequences together?
For clustering, we basically use like a blast value of 10 to the minus 3 as our most coarse cutoff. But we know that if we build different size alignments, we get the information changes slightly, and it can become more specific to particular subfamilies or less specific. So that's something that I'm trying to understand more about. But yeah.
Have you participated with this method in the CASP competition by any chance?
No, I haven't. I don't really. CASP is very much. We'd have to refine these structures basically, and that is sort of a research field in its own right. So I guess we get this sort of slightly rough structure, which I think is really useful. I'm not sure how much more useful it would be to. Sorry.
Just trying to see whether these need to come up or.
You had a set of globular proteins. How did you choose that set?
So I chose proteins for which we had, I think it was a minimum of like 500 sequences. I wanted proteins of different sizes. We picked them randomly basically from PFAM families. So, yeah, there were enzymes and there were other non-enzyme proteins, and it was just chosen to be sort of representative, basically. But we haven't published all of those. Yeah. Okay, thanks. Yeah.